A very good morning, everyone. On behalf of the CSTEM co-chair team and Danforth Center, I would like to welcome Dr. Joanna Porankiewicz-Asplund from Agrisera. And also I would like to welcome you all for your active participation in this workshop. So before I get into a few logistics here, I would like to thank Bill for making this collaborative workshop between Sweden and St. Louis, United States possible. So thanks, Bill, for that. With that, I would like to introduce Dr. Joanna Porankiewicz-Asplund. She has graduated from Adam Mickiewicz University in Poznan, Poland, and worked with heat shock proteins in lupin roots. After 3 years of postdoc at Umeå Plant Science Center, she joined Agrisera. The company is an antibody supplier from northern Sweden. Over the last 20 years at Agrisera, she was developing antibody purification methods and troubleshooting different antibody techniques, as well as she is been involved in product development and educational activities. So without further ado, I would give the floor to Joanna. And I think in doing all this, I forgot to introduce myself. I know that most of you are aware that my name is Thea and I am a research scientist in the Allen Lab. So the floor is yours, Joanna.
Thank you very much, and welcome everyone to the largest workshop I've held online. And just a little bit of technicalities, because we have 2 hours, but it is quite a long time, and the attention span can be a little bit exhausted. Therefore, there will be a break in the middle for you to grab a cup of tea or coffee. And also there will be 2 surprises, one in the middle, and the second surprise will be in the end of the workshop. And if you are not comfortable with asking questions during my presentation, you can write them in the chat and I will address them in the end of the workshop. Please don't be shy. It's nothing like a stupid question. There are no such questions which are stupid, because every question has a reason. And if I can help you to improve your Western blots and share my knowledge with you, I am more than happy. So you can either interrupt me during my talk or we do it later. And you can also send me a message later by email. I mean, anything is fine. And now I will share my screen and we start the workshop. So, Western blot, a technique possible to optimize. This is a workshop for the Danforth Center. As Western blot is quite a complex technique, I would like to share with you my knowledge. Over the last 20 years, I worked with Western blot, and I have seen hundreds of blots, and I was helping sometimes very exhausted scientists to make this technique to work. Based on that, I prepared this material, and I hope that you can both listen to this workshop recorded, but you will also receive PDF of this presentation later. And on some slides, there will be links to different documents where you can deepen your knowledge if you need it. Agrisera, where are we located? First, we start with a little bit of geography. So as you see here, we are a little bit like on the same latitude as Alaska, which means that in the winter it's going to be a little bit of snow, and sometimes we have problems to get to the office, and we have a winter day in the winter. Because of our latitude, there will be mainly dark. And in the summer, we have a summer day. So one can garden whole night in Sweden. This is where we are working. And this is where Umeå Plant Science Center is located. And we have very strong collaboration with them also in antibody development. And Agrisera is also ISO certified company and is located 1 hour flight from Stockholm. And our antibody production facility is located out in the countryside. It's very beautiful there. And we have also a facility in Umeå where antibodies are purified, where we do QC, where we run ELISA and Western blots. The company was established quite a long time ago, so 1985. We are now part of Olink, a diagnostic company, but plant antibody unit is running as a separate entity. At the moment, we have 45 employees. Plant antibody unit is 7 people. Western blot, it's a very useful technique, which actually came to life quite a long time ago. And this has started, you see, 1979, when already this idea came to life. So you're actually working with an ancient technique, one can say. The base of it is a sandwich concept, which has been also used in other methodologies. So you're probably familiar with Eastern blot, Southern blot, which is for DNA. We have Northern blot for RNA, and we have Western blot for proteins. And the name actually comes from, it's a little bit of a joke. It comes from the lab which was placed on the Western bank. And the original article which described this technology, the Western blot ID, has received quite a lot of attention back then. And it is a concept of a sandwich we are working with, and it's important to understand how this works to be able to troubleshoot it. And I know that there are experienced users between you, and there are also beginners. So I will try to explain it. For experienced users, it will be a reminder. For beginners, it may be something new. So we have a transfer stack with, this is blotting. We're talking about blotting here. So we have the sandwich where we are applying a current, and due to the current, the proteins which are separated on a gel are moving to the membrane. And of course, we have filter papers and a sponge. And we apply the current, and we make the proteins to be blasted from the gel to the membrane. So the purpose of it is to obtain an exact copy of a gel on a membrane. And this is in a way genius, because this will allow to target a unique protein in a very complex mix, which we are separating on a gel. Who is doing that job of protein identification? That's an antibody, and an antibody molecule is in my background. Schematic representation is like you see here. We have different antibodies. Llama antibodies are a little bit shorter, but the antibodies which you're mainly working with are coming from different mammals. And we have so-called constant region, which is not changing. That's an important part. And then we have the most crucial is the antigen binding domain, which actually is accommodating a binding site to your target protein. And the result of it should be like that. Antibodies binding to a target protein, and I will not go into details of polyclonal and monoclonal antibodies here, because they are binding in a similar way. Just a reminder for you that poly means many, so it will be antibody pool recognizing different binding places, while mono is a single. So this antibody will recognize a single binding place. That's the difference. And there are different advantages and disadvantages of using either poly or monoclonal antibodies. So please remember that in this antigen binding domain, this is where the binding to your target protein occurs. And if we zoom in more exactly, then we can see that in the antigen binding site, we will have at least 3 amino acids in linear sequence. This is where the antibody will get attached. Or the antibody can also recognize a 3-dimensional epitope. So as you can see here, there will be separate amino acids, often located in distant parts of the protein, which will be brought together by protein fold, and in that place the binding occurs. So in Western blot, we're mainly working with linear epitopes, which are made from subsequent amino acids, and an epitope can consist from 2 to 15 amino acids. Polyclonal antibodies will recognize different epitopes. Monoclonal will bind to only one epitope. Therefore, sometimes monoclonal antibodies are only useful for one technique. Polyclonal antibodies are more versatile. They will bind to many epitopes. So please keep in mind that in Western blot, we're mainly working with linear epitopes. Western blot in denatured conditions, protein is unfolded, antibody binds to a linear epitope. And then it will look like this. Oh, I should remove that bottom panel so you can see more. Or I do like this. So antibody binding will remind a pyramid, or we will have a target in the bottom. We will have primary antibody, which recognize our target, and through antigen binding domain binds to a target. And then how do we visualize this binding? We do it through a secondary antibody, which needs to be recognizing the primary. And the secondary antibody has some kind of label. It can be enzymatic or it can be fluorescent, and therefore we can record that binding. It may be a little bit complicated, but if your primary antibody is made in a rabbit, then the secondary needs to be anti-rabbit. It's logical. Sometimes it happens that people may use a wrong secondary antibody, but this is not such a common mistake. As you see here, we also need a blocking reagent. So blocking reagent is simply, as the name implies, is used to cover up all the other sides. Of course, it's a random process. But antibody will anyhow penetrate and bind to the desired target. And there are two different types of this detection method possible, because it's also one where primary antibody can be labeled. It's not so often used, but this is much simpler and faster detection. However, more useful for proteins of medium or high expression. So, for example, photosynthetic proteins most often are quite abundant, and therefore we could use directly labeled primary antibodies, which will shorten our protocol considerably, because we would not need an incubation with secondary antibody. So we have two types of the detection possibilities, either through secondary antibody, which also makes an enhancement, or directly through the primary antibody, which is marked. And that's that label which makes it possible for us to detect, visualize binding with the target. And when one looks at Western blot protocol, you can get quite overwhelmed because there are quite many steps and quite many parameters. And I will walk you through those steps, and with the knowledge which I have obtained troubleshooting those steps in Western blot, I will try to show you what to pay attention to. Because usually we run to the lab and start producing blot after blot, but actually we should stop and think first. And I will show you why. So theory is good. How about practice? On this slide, you can see 4 different labs which received exactly the same primary and secondary antibody. And they returned to us these results. So with the same reagents, the material was Arabidopsis thaliana grown in those laboratories, but the results, as you can see, are quite different, from no signal to a lot of background to actual visualization of a target protein. So what was different? The protocol was different. The antibodies were the same. So how much do you know about the protein which is to be detected by an antibody? This is a crucial thing for your success. Antibody needs to bind to the target. I would like to show you a protein checklist. What do you know? Do you know anything about biochemical properties? Is it a membrane protein, or is it a cytoplasmic protein? What expression level your target has? There are different databases where one can obtain this information. Do you know organ localization? Cellular localization? All this has to be studied before you go to the lab. And believe me, this will save you extreme amount of time. I know that you like to probably to do experiments, but if you do this theoretical preparation, it will save you a lot of time and pain later. Target protein sequence. How do I choose the right antibody for my protein? Should I know about origin? What should I know about origin of my protein? I built this workshop based on questions which I received from you and during other interactions with people doing Western blots. So look at this sequence. The antibody can be made either to a whole protein, or it can be made to short synthetic peptides, which makes the antibody monospecific. So antibody can target N-terminal part, C-terminal part, or in middle of the protein. And if you are using an antibody from someone, you have received it, instead of going and running a Western blot, you should first check if the antibody you have received is actually made to antigen, which you can find in a sequence of a protein you are working with. It has happened many times that people omit that step and come disappointed. It didn't work, I have no signal. Yes, because maybe this underlying amino acid sequence was not found in the protein they were working with. So the sequence homology has to be at least 70% for the antibody to bind. And of course, we're talking here about linear sequence. We're not talking about these 3-dimensional epitopes, because those are unknown to us. But we can at least predict if a given antibody is going to work or not. So if you are planning to use an antibody from a company or from your colleague, you will really save yourself a lot of time if you do this check. And these predictions are very accurate. And we have had cases when people purchased the antibodies and came back to us later and asked for this checking the sequence, provided us with sequences. And then 3 antibodies out of 5 were working. And then I sent the prediction result, and he said, yeah, exactly, this is the experimental data I got. So for the other 2, there was no conservation. Therefore, there was no positive result in Western blot. So remember that this is a homework to make, to check first if there's conservation between the sequence antibody is made to, and if that sequence is found in your target protein. Amount of target protein in a tissue are very useful databases to check that. So for Arabidopsis, we have AX database, which will give us approximate amount of target protein per tissue based on experimental data. And my favorite is ePlant from University of Toronto. And that database is not only for Arabidopsis, there are many crops and edible plants in that database. Of course, mRNA data may not correspond strictly with protein expression, but this will give you at least a little bit of guidance if there is a tissue where this protein is expressed. Is there maybe a developmental stage? So maybe it's better to analyze senescent leaf, not a young leaf, at least in initial stages of working with an antibody. So it's very important to actually check this up before doing Western blots. A protein extraction method is also something which we have to consider. And I will show you why. Because if you see in this example, when total leaf extract was used, there was no target protein detected. There was no band. But with using specific fractions, the band became concentrated. I mean, the target protein became concentrated, and therefore detection was possible. So in total leaf, target protein was too diluted for the antibody to be able to find it. Once specific fractionation has been done, the target has been concentrated, and then visualization was possible. So for proteins which are not cytoplasmic and are of lower expression, it may be very useful to fractionate. I know that fractionation is not so popular because it requires time, but believe me that it is very useful to do it. And I think many antibodies have been discarded because people use them on a total extract without fractionation and got disappointed with a negative result. As you see here in the first lane, there's no signal, no band. Also, if you wonder why this part is changing, it is because by fractionation, some proteins will fall off. So the background pattern will also be different. And this result, which I present here, can be also optimized further. So by blocking, because Western blot is about signal to noise ratio. This is what we are after, to achieve the best signal to noise ratio to record a target protein faster than any background will show up. Part of protein extraction method is also to consider buffer composition. And we actually have on our website results like this one. Depending upon the most commonly used is, of course, trees, but this is not the most optimal buffer for protein extraction. Some proteins are not sensitive for it, but while the others may require actually heat, which buffers a little bit better. And this is an actual result from one of the labs in the US, which have been testing different extraction buffers. And in buffer 1, there was no target band. In buffer 2, the target band appeared quite well. Extraction buffer in some cases has to contain urea, which is 6 to 8 molar. And this is also a result from one of the labs, Arabidopsis. And then we had 2 crops, rice, and there was no signal at first, unless urea was used, and then the target protein was denaturated further, and the target band was detected in the other species as well. It is the same protein can actually have probably a different fold or interaction with some cofactors, and that makes can make binding of an antibody more difficult. Therefore, such a result. You should also consider specific organ. We actually had a case when with PIN2 antibody, if a whole root was homogenized, there was absolutely nothing, no detection. 2 millimeter of a root tip had to be used for extraction, and then the band appeared. Of course, protease inhibitors are a must. You may be surprised, but sometimes people send me protocols without protease inhibitors included. And therefore, if extraction is not done promptly, the target protein will be simply degraded. In some cases, proteasome inhibitors have to be included. Why is that? Some proteins you're working with may be actually a target for proteasome degradation. And this is a story from a lab which was for 1 year trying to detect their target protein, and the blots were just blank until someone in the lab got that ID, because actually their target protein was a target for proteasome, and therefore they couldn't detect it. Following these links, you will find a little bit more of information. I got a lot of questions about the electrophoresis. This is my favorite apparatus. It's a Novex system from In Vitro Gen. We've been working with it for 20 years, but there are plenty of other ones which will also give you good results. What things should we take care of during SDS-page and electrophoresis? ELP means good laboratory practice. So what is good laboratory practice? Clean. I mean, you need to be really, really clean. And for some antibodies giving very high backgrounds, it can help to change, for example, containers you incubate in. Of course, cold. You need to work in the cold. All the smiles during electrophoresis are due to the heat. So cold, clean. What more? Need to be precise. I mean, careful. You need to also have fresh reagents. No shortcuts here. You need to follow the protocol, and this hopefully help to be also consistent, because this is what Western blot is about, we need to be consistent. So remember, clean, cold, and absolutely fresh reagents. So think twice if you're going to reuse anything, and these shortcuts can cost you your results, especially if you're working with very precious samples. I received the question, what should I load per well? I recommend microgram of protein or microgram of chlorophyll. In very many publications, many labs are loading on volume. And why is this not good? I mean, it is very difficult to compare such results if you load on volume, because different extractions made maybe the same week or the same day will not yield the same microgram of protein per microliter. And it is like going in the darkness, in my opinion, to load on volume. There is an argument which I often hear that one cannot measure protein in loading buffer or with denaturation reagents, with DTT, detergent, and so on. That's not true, because there are reagents which are compatible with detergents, buffers, salts, anything. So it is really possible to measure protein concentration, and I strongly recommend that for your good Western blots, you should actually do it, because it will be easier for you to optimize them if you know protein concentration of your sample. Protein load per well. How much material should I have? Nanograms or micrograms? It actually depends. Depends if you work with what is expression level of your protein, how much of it is in your extract. And here you can see titration experiments. So of course, if you dilute the primary antibody, the background may also dilute and be lower. If you titrate the protein load, then you will also change the proportion with the potential background. But this is the action which is to be done once you already have your target band detected. So here I'm giving you an examples how to refine your Western blot. Sample denaturation before we load anything on a gel. In denatured condition, the sample should be denatured. And what you probably have seen in different protocols is a range of different temperatures. As you can guess here, we are fond of 70 degrees for 5 minutes. We are not fond of boiling, extensive boiling. The reason for it is very simple, that when heat treated, proteins can aggregate, and the aggregate will not enter a gel. So the target band is not going to be detected because the protein is stuck at the top of a gel. A classical example is H+ ATPase, which aggregates in heat. So 5 minutes at 70 degrees is our recommendation for sample denaturation to avoid that issue in the future. Gel electrophoresis. I got a question about the formulation because there are different gels available, 3-styrene, 3-glycine, 3-acetate, and one can feel a little bit lost what to use. Or maybe you are casting gels yourself, so why using precast gels? I mean, they cost. Molecular weight range will be different for different gels. So this comes back to the question of how much do you know about your target protein, because this is what decides what happens later. Some gels will have different pH stability. They will have different shelf life. depends what you're after. Some gels can be used for over the year. The others will deteriorate when stored in cold, depending upon their composition. Also, you can get better resolution. Here, I am showing you thermofusion data for high molecular weight protein, which was separated in different type of gels. So 3-glycine versus 3-acetate. So the picture says more than 1,000 words. You can get better results if you adjust the gel type and oftentimes also a buffer system. When it comes to precast gels, I'm actually fond of them because we have been doing quantification work, and the most consistent results were obtained with precast gels. It saves time, it saves labor, and there is a bigger variety to choose from. So depending if you maybe switch your project and you start working with protein of lower molecular weight, then it's better to use another type of gel. I mean, for result consistency, these gels are really, really good. Casting gels yourself is often happens in uncontrolled conditions. So this will also influence your results. Sometimes it may stop working. You may use a lot of time to troubleshoot. So precast gels are a very good investment, which I would recommend to streamline your Western blots. Protein transfer conditions. Here, what to keep in mind. I got the question, is it better with semi-dry transfer in a cold room, or what should I think about when I'm doing protein transfer from a gel to a membrane? Think about the gel as a net. So depending upon the percentage, the net has different eyes. I mean, the big proteins will get stuck at the top, smaller ones will migrate further down. And you can play with gel percentage, and in precast gels, you can get quite good combination in the gel percentage, which will give a good separation for high molecular weight or low molecular weight proteins. Of course, work in the cold. And as you see here, depending upon molecular weight, you have to do certain adjustments because big proteins will leave the gel slowly. Small proteins, it's a risk that you blast them through. So you cannot use the same conditions always for each protein because the lab has done like that for the last 5 years. You have to do your homework, the theoretical work first to succeed later. As you see here, we recommended is a bigger pore size of a membrane for high molecular weight proteins. And also one can play with methanol and SDS. As you see here, for high molecular weight proteins, a wet tank transfer is actually recommended. It works better. So low molecular weight proteins will be blasted from the gel faster to the membrane. High molecular weight will need more time, and therefore you need to keep in mind that you need to adjust the next step in the Western blot, but you can do it before you go to the lab. I received the question about which method is better, wet or semi-dry system. When it comes to amount of buffer, of course, wet uses more buffer, and semi-dry uses less buffer because there's almost no buffer there at all. Electrode contact is through the buffer with wet transfer. It's a direct semi-dry transfer. Speed, as you can guess, will be faster with a semi-dry, but faster is not always better. And in semi-dry, there will be more risk maybe for air bubbles So an ideal situation would be to have two possibilities in the lab and see which one works better. We are using wet transfer with very good results for proteins of quite big span of molecular weights. But of course, nothing is written in stone, and it can work for certain protein. It can work better with semi-dry transfer compared to wet transfer with a certain protocol setup. There's also one thing to add here. Wet transfer is less prone to failure. So this is maybe to start with. And remember, faster is not always better, especially if you work with small proteins. You can completely blast them from the gel through the membrane, and they can just be blasted to the buffer. So you will not capture them, and you may be convinced that antibody is not working. There are two different types of gels. We have 1D gels and 2D gels you probably heard about. So how the dimension of gel affects the results. When you think about 1D gel, it's just bands, right? Like here, you can see here. But each band can actually contain quite many proteins. So the 2D gel will allow you to separate proteins which are in that single band, which you see in 1D gel. And it depends what questions you're asking. And there are a little bit of differences in separation, depending upon if it is 1D gel or 2D gel. So there are no limits in PI in 1D, while there are limits in PI in 2D. So PI is electric point. And also 1D separation is more suitable for membrane proteins. And 2D has a certain kilodalton limit, but the methodology to work with them is similar if you want to use antibodies and detect target protein afterwards. Protein transfer, which membrane to use? And it is a very interesting question here. Sometimes labs have their favorite membrane they are using, but for certain proteins, one type of a membrane can be actually better. So if you see here, VDF membrane binds proteins through hydrophobic interaction, while nitrocellulose hydrophobic and electrostatic interaction. But there are a little bit of advantages with PVDF because it can be air dry and rewetted. And why would that be an advantage? You may ask. This will cost me more time in the lab. But actually, if you dry PVDF membrane, think what happens. The protein may unfold further. So the accessibility for the antibody will increase. Also, PVDF membrane is more robust. So, for example, we are preparing our membranes in advance. Yes, so instead of storing precious extract in a freezer where it can degrade, we prepare the membrane, we run a gel, we do the blot, and then we store PVDF membrane in room temperature for up to 5 months. And we catalog what we have separated, what extracts we have transferred to that membrane, and we can use it when we need it. So, of course, I'm not going to discuss which membrane is better because it depends upon your target protein. And for many antibodies, many proteins, equally good results can be obtained with both membrane types. Of course, there will always be exceptions. So if there is an antibody which we know works better with nitrocellulose, we will write it in product information sheet. So that information will be there to help end user to limit amount of problems with their Western blot to be more successful. Then when we do protein transfer, it is very good to do quality controls. I mean, this is a must, especially when you are a beginner. What is a quality control? After transfer, you can assess your transfer quality by using Ponceau-S. Reversible stain can be very fast removed from the membrane with water. And of course, we wouldn't recommend to continue with the membrane, which looks like the one you see here. It's also saving your time. So I wouldn't continue with the protocol, I would stop there. And the second control, so here we want to see if proteins have been actually transferred to the membrane and how the quality of the transfer is. And the second control is if proteins left the gel. So you simply stain the gel with Kumasi. And why I brought up this slide, this is a double sandwich. So we have two gels. And as you see here, gel 1, when stained with Kumasi, has no protein. Okay, everything went to the gel, to the membrane, and there is a copy. But on a gel 2, proteins were left. So if you work, your target protein is from molecular weight range, as on this gel, there is a risk that it has not been transferred completely. We have been doing quite a lot of quantitative work, and we decided not to use double sandwich because of that reason, because then our target protein is not completely transferred to a membrane. For initial work with characterizing an antibody, one can use a sandwich with two gels. But later, if you want to do any quantification, I would like you to be aware that it should be good to check if actually there are no proteins left behind in a gel. Primary antibody format, what is that? You're probably familiar that antibodies can be provided in serum or they can be purified. And there are two different types of purification. One is done by protein A or G. Antibodies are attaching to this handle. It means that all antibodies to all antigens are pulled out. But we have so-called antigen purified antibodies. It means that antibodies passed through a column where protein or peptide an antibody is made to is coupled. And be careful when you're buying antibodies, because these both types of purifications are called the same because it's affinity. But there is a difference what you get afterwards. And here you can see the result of this affinity purification. So if you purify an antibody to total immunoglobulin, it's only a small fraction is going to be against your target. If you purify an antibody on the antigen column, then most of what you have in a tube will be against your target protein. An amount of specific antibody in serum is unknown unless we do specific antigen purification. And this is how the purification is done. It is simple, it looks, it is straightforward, and this can be done also just on the bench in case you ever need it in the future. So first we need to couple peptide or protein, which was used to make an antibody to a resin. Then we pass serum through, and then specific antibodies are going to bind, and they are eluted, usually by low pH, which makes conformational change of the antigen, and antibody is released and collected. We are using a chromatography system to purify our antibodies, and we are using these small columns. Of course, the column bead can be larger depending how much of antibody we need to purify. And this is a very interesting question which I received, and experienced users are probably very familiar with it. how long can the same batch of antibodies be used, so reused. And some antibodies can be only reused one time, while the others can be reused 20 times. And if you're working with not characterized antibody, I would definitely not recommend you to reuse it. Why is that? Because it depends upon how much of antibody to the target is in your preparation. And some antibodies will bind in the first incubation. Most of what you have applied on the membrane will bind, which means that if you reuse the same solution in a second experiment, there will be no primary antibody to bind. So there will be no reaction. So this has to be determined experimentally, but I wouldn't recommend it in the initial steps of working with an antibody which is not fully characterized, which you received from someone. And if you don't have a signal, once you tried to reuse it, it's not an antibody fault in that respect, that you simply cannot reuse it. It is one-time thing for some of the antibodies. And here we reached the page.
Joanna, this is really good. I'm enjoying this. I understood nothing about it until you started explaining it. I kind of got lost. I got distracted because my assistant came in. He's working on some stuff as well, but breaking this up into chunks would be perfect for a knucklehead like me, because I don't follow as well. I don't have the background information, but I've been understanding pretty much when I can tune in. I've been understanding everything that you've been saying. So this is really cool, and it's been kind of revolutionary for me, because like, oh, I didn't know that's how that worked. That's cool.
Oh, that's nice, because that's I think it's an art to explain complicated technique in a way that it is comprehensible, because this is, you know, people spend hours fighting with the blots and trying to get good results. So it's thank you that you're saying that. I hope the other participants will have a similar feeling, that for those who are experienced, that they will also find something useful.
Well, I was going to say, most everybody on this talk is way smarter than me when it comes to this stuff. So I think they're probably getting more out of it than I am. But this has been really cool. I'm enjoying this a lot. So I'm glad I got a chance to sit in on this one.
Oh, thank you very much. Oh yeah, thank you.
You're welcome.
It looks like we have 3 questions, including one from me. Would you like to read the question yourself, or you want me to do it? Either way is fine for me.
All right. Yeah, I can read the question. I hope I found all of them. Does this fractionation also necessary in case of nuclear proteins?
Yes, that's one. And then you already addressed the difference between the nitrocellulose and PVDF membrane. So we should be good on that. And then the continued is about cleaning the membrane.
Yeah, I saw that one.
Yeah, and I actually have one question which I added there as well.
Okay, should I start with the first one, fractionation?
Yeah, that's fine. Yeah, when I hope everyone will get back, and I just want to remind, I will wait for like a minute, and then I will remind everyone to turn their cameras on if possible.
Okay, and so I addressed the first question. Oh, 2 new messages, great. Does this fractionation also necessary in case of nuclear proteins? I say like this, if it is transcription factor, yes, because these are usually not so abundant, so you need to concentrate your target before you can detect it. If it is an abundant protein, I mean like histone antibody will work without problems on a total cell extract. But if it is the low expressed protein, absolutely nuclear fractionation will improve your results. I've seen blots where from no band, there was a band. So this is a proof of concept in that case. Please remember that you need to make this theoretical work. So the more you understand about your target, the better you succeed later. Cleaning.
Joanna, I'm just sorry for interrupting you. So my question is, if the nuclear protein is somewhat overexpressed, that means you are making transgenic, and the protein is going nuclear. So still you need the nuclear fractionation, or it will work?
It depends upon overexpression level, because if a protein is overexpressed, it can be quite strong overexpression, then you would not need to do it. But sometimes if it is not so successful, you may need it. So you need to experimentally determine that.
Yeah, because we can see in the QRT like PCR that okay, the genes are expressed, but now when you see the proteins, oh, you can't see the protein. That means, do you need the nuclear fraction, or they are not working at all? How you will decide on that?
Well, if you see that there is an expression but you can't detect the protein, I would definitely try to purify nuclear fraction. It's not that complicated protocol.
Yeah. Okay, thank you.
You're welcome. Cleaning membrane after gel transfer to remove leftover residual gel particles. I've never had residual gel particles on my membrane, unless you maybe, if you are not gentle enough, and because membrane has to be handled with highest possible care. And you need to be careful when you disassemble your sandwich, for example. And I have never seen such a situation. I would never scrub my membrane. Absolutely not. I use forceps. I transfer it, holding it just in one corner. I will mark where is the protein side, because that can be important. I will use gloves. I mean, think like you would be working in a really careful environment, because absolute no scrubbing, no physical force on the membrane. Can stain molecular marker transferred serve as QC? No, because the mark. Yeah, I would like to see still a whole membrane.
Yeah, this was the question I was having, because, you know, you mentioned that you can, after the transfer, you can stain the membrane with Kumasi to see if there's any remaining protein. But for example, if in one of the, if we load a molecular marker, which is pre-stained, and then we transfer the gel to the membrane, everything will be transferred, and we can see all the marker stain in the membrane, and it's completely empty in the gel. Is it not enough, or is it still a good idea to stain the gel with Kumasi?
Well, because, you know, the electrical field, it's hopefully the same in all parts of the membrane, but may sometimes not be the case. And this staining QC is quite a quick procedure, so I wouldn't rely. We also use markers which are stained and by color, and we mix them with so-called hot markers, magic marks from Invitrogen, which will give us molecular weight during ECL development. And this is very helpful, because then you get identification of molecular weight very correctly. But back to your question, I would still do a quick stain of the gel with Kumasi to see everything has left the gel. Is it okay? Because if there's some issue with your apparatus, you will be able to identify it.
Okay, thank you.
And then we have PVDF versus nitrocellulose. What property of the protein should be used to determine between the two? We had experience of hydrophobic protein like PSAA, photosystem 1 protein, which was working better on nitrocellulose. than on PVDF. But as I said, for majority of proteins, both membranes seem to be equally good, at least for those proteins we were working with. However, if you have both membranes in the lab and, for example, experience background issues, you may try to test the other membrane, just like a quick test, if there will be any improvement. Have we exhausted all the questions now, or have I missed?
Yes,
something?
I think so. I think so. Yeah.
Right.
Just one quick reminder to all the participants, if you can put your name and lab name in the chat box, and if you think you can turn the camera on, that will be very nice. Thank you.
I hope that for those who joined the quiz, that you had fun. There will be one more chance at the end of the workshop. So let's move on. We are coming now to we are moving on in our Western blot protocol, and we are coming to membrane blocking and background, which is quite a common issue. And why is that? We are after the best signal to noise ratio. Please note that unless you really overload your system with too much of everything, for antibodies to proteins of high abundance, usually background is not an issue. And we have different blocking reagents. So BSA you recognize. We have non-protein blocking, PDP-40, used also for fractionation. Most commonly used is 5 to 10% non-fat milk. I've seen the best results with non-fat milk. Somehow for plant extract, BSA seems to be too weak. This is the experience which we have seen over the years. Blocking, as you maybe remember, is an important part of the procedure to cover everything up for the antibody, so the antibody can just target the epitopes which we wanted to bind to. We are also having a possibility to block with gelatin, and we can also use commercial blockers. I have seen labs using gelatin, but the results with gelatin were not so good, but it will also depend upon antibody protein pair and upon the extracts you're working with. So I'm not saying that something is obsolete, because there are many possibilities. I can only guide you that in our hands, that non-fat milk seems to be working very well. There are also a range of commercial blockers which have proprietary recipes, which can also be quite helpful for some kind of difficult antibody protein pairs. And here you can see an example of what the blocking can change. The lab reported that, look, we have many bands, this antibody is no good. But as you can see here, they blocked only for 30 minutes with very low percentage of milk. While adjusting, extending blocking and using milk at 5%, suddenly the antibody is working well. No, it's not antibody working well. It's that the protocol is optimized. So the signal to noise ratio, which is obtained, is much better in this case, because there was a possibility to block off non-specific binding places. For some antibodies, blocking at room temperature is enough, while for the others, we have to block longer, and then sometimes it has to be done overnight. As you can see in this example on the second half of the slide. So usually when we work with unknown antibody, we will block with higher percentage of milk and for 1 hour, incubate primary antibody overnight, and depending upon the result, we adjust it later. In the initial experiments, the most important is to see a target band being detected. So we worry about the background later. How to reduce background signals while stripping and reprobing. Personally, I'm not so fond of stripping and reprobing. And why is that? Why background signal can increase when you strip and reprobe? Why do you think is that? When you're stripping, you are also stripping part of the target, because this is quite a harsh protocol when we are stripping. So when stripping and part of the target is also stripped away, the signal to noise ratio will be shifted towards the background, because there will be less of the target for antibody to bind. So I would suggest to use higher percentage of a blocker, for example. If you are doing some kind of initial experiments and you absolutely need to do the stripping and reprobing, this can maybe help. But if you're doing quantification work, or if you're aiming for results to publication, I would not recommend this procedure. Because keep in mind that by stripping and applying these harsh reagents, you will be breaking up the bond between the antibody and the target, but also releasing some of the target from the membrane. Comparing primary antibodies, does it work? I work with not fully characterized primary antibody to protein of low expression, and I noticed that I get the signal with actin, but not with the primary antibody. Why is that? We're here still about the signal to noise ratio, because we cannot compare actin, which have high abundancy, to a target protein of low expression, because signal to noise ratio will not be the same for those two proteins. So actin is often used as a loading control, and this is correct. But when characterizing an unknown antibody, or we have received an antibody from a colleague, we should be careful and try to optimize the protocol first towards that target protein. There is also, if you are purchasing antibodies, you should receive information from a supplier, what conditions to use, and please study the protocol first, because that's the most common mistake people do, is that they will use the protocol which they always used. Instead of looking at product information sheet, there are often pictures with description, I hope, and there you can get your parameters. So stick to that, and also check how the antibody should be stored. That's just my sidetracking here, but it's an important information for you. Antibody incubation buffers. I think most of you is using probably PBST. But here I would like to show you that there can be, for some protein antibody pairs, an improvement when using PBST buffer. This is an example of experiment which was done with our catalase antibody, and why is that, I unfortunately cannot explain. I just would like to flag it for people who may have some extensive background problems, that this slight change can make a difference. For many protein antibody pairs, it may not matter. It is only for certain combinations. And now we are coming to an issue of saving time, and that's maybe something what is important for you, and therefore I brought it up. There is a possibility to incubate several antibodies together. You maybe have been not doing it yet, but it works. There are many labs which are using it. So instead of running 3 plots and spending 3 days to get the result, they will use 3 primary antibodies at the same time. Of course, this is recommended for antibodies which you know how they work. And also, as you see here, there has to be at least above 20 kilodalton in difference between these target proteins. But believe me, this works and this saves your time. Another possibility to save time is to incubate primary, secondary antibodies mixed together. And there is a reagent called IncuBlocker, which Agrisera offers. And here you can see an example that you can get the reaction already after 30 minutes. So think how you will shorten your Western blot protocol. You don't need to use IncuBlocker. You can use your primary, secondary, and blocking at the same time. So think how many washes and steps it's going to be saved. But be careful here, because this works for proteins of high and medium expression. If you work with transcription factor, this can be a little bit more problematic. But maybe worth to try if you are to do a lot of Western blots with a well-characterized antibody. And a third method to save time is to use directly conjugated primary antibodies. And they can be conjugated with alkaline phosphatase, with horseradish peroxidase, so enzymes, or with dyes like this fluorescent dye light. So in that setup, secondary antibody incubation is not necessary. This I would also recommend for proteins of high and medium expression, because for proteins of low expression, the enhancement is given by adding secondary antibody, which will bind in several places to a primary. So there will be an enhancement step. But there are 3 methods to help you to save time with Western blot. And then we come to secondary antibodies. Here you can see that it actually may matter which secondary antibody you're using, because some are better than the other. They differ in dilution, they differ in cross reactions, and they differ in incubation time. Incubation time should be always short with secondary antibodies. Please keep it in mind. Do not incubate for more than an hour, because you can overload the whole system. And usually people are very concentrated in price of antibody, considering milligram in a tube, and then the price tag. But that's wrong, because what you should look at is a dilution, because there is a big difference in an antibody which you can dilute 1 to 25,000 times, and the antibody which you can dilute only 1 to 5,000 times to obtain a result. So please look at dilution, because secondary antibody which can be highly diluted will last many more experiments. And then we come to detection methods, and there are 3 detection methods which I will present here, which you are probably familiar with. First one is a chemiluminescent detection. So we are using a CCD camera to record it, and we have an enzyme conjugated. So an enzyme, horseradish peroxidase, is conjugated on a secondary antibody. And here I would like to show you the result with 2 different ECL reagents. These are the reagents which are a price. So we have a sensitive reagent, ECL super bright, with extreme low femtogram detection range, and when we have ECL bright, which is having a medium detection range. And we applied it on to detect PSBA, a photosynthetic protein of photosystem 2. And as you see here on Blot A, one would say, oh my God, so much background. This antibody is really bad. No, it isn't. It's that too sensitive detection reagent was used. So remember, we are after signal to noise ratio. So for high abundance proteins, to detect them and visualize, one does not need to kill the system with super sensitive ECL detection, because this will result in more background. This is also important to keep in mind when you're choosing your reagents to visualize antibody to target protein binding. And here a question: why do we see ghost bands sometimes? What causes them? So ghost bands are appearing on the plots in cases of secondary antibody excess. So there is too much of secondary antibody, and the reaction is going so quickly that instead of recording a band, we will see just the white glow. Sometimes it's also, there can be an antibody mismatch. What do I mean by that? It is a rare situation, which I have seen in the past. It happens maybe a few times per year, that actually primary antibody and secondary antibody will not match. So to solve this is maybe to go to the lab beside you and ask if they have another secondary antibody from another supplier and see if this would be better. But usually, in most of the cases, ghost bands are when secondary antibody is used too concentrated. So the product information sheet said 1 to 25,000, but to be on the safe side, you used 1 to 5,000. And this is way too much. Another question concerning detection with ECL is if the blot does not develop properly or something went wrong during documentation. Can the ECL incubation be redone? Can one redo the same primary and secondary antibody without stripping? Answer is coming, yes. Do it within the same day. So you can still save your result and redo just the last step. Second detection type, which is more and more popular, is the fluorescence. So we have a secondary antibody conjugated to a fluorescent dye. And here you can see actually simultaneous detection of two different targets with different secondary antibodies. So one is conjugated to a dye which is kind of green-yellow, and the other to a dye which is red. What I have seen is that many labs have both possibilities. So if one fails, they will move towards the other. And I have also seen that sometimes there is a mismatch. So secondary antibodies provided together with the equipment used for fluorescent, for recording of fluorescent reaction do not match with the primary. So to solve it is to change the secondary antibody. In terms of sensitivity, ECL and fluorescent detection are considered to be more sensitive for at least Western blot technique we are discussing here. The third type of detection is using colorimetric detection. So we have actual reaction with its product visualized on a membrane, which can be used in a lab book, for example, for documentation. And in our hands, this type of detection works well, but still, if you work with proteins of low expression, ECL or fluorescent detection can yield better results. So here we have alkaline phosphatase conjugated secondary antibody, and we do not need any digital camera. This is an advantage of this type of detection. How to get an intense signal from Western blot using a weak antibody? Well, one has to first analyze your protocol, because it's usually antibody which is blamed directly. This antibody doesn't work. I fight with it for weeks and does not yield the results. Instead of spending time in the lab, go to the bench and start analyzing. What sample type do I have? Is a fractionation necessary? Because it may be. What protein load per well do I use? Do I measure protein concentration? If you do, then you don't know. Do I have optimal transfer conditions? Or do I use the protocol which was always used in the lab? I work with protein of 120 kilodalton, but I use the protocol for low molecular weight proteins. Do I use purified antibodies or is it serum? Do I use appropriate detection reagent? So all this is called optimization. And I would really like you to get the home message from this, that doesn't mean that you have to multiply the conditions and test all this. No, you should think about it before you go to the lab and plan your experiment on the paper, consider all these factors, and afterwards go to the lab and do a Western blot. Some examples of optimization. What was changed here? Such a trivial thing as protein load per well. Before, it was used too much of protein, and therefore the background. When this was adjusted, the result is much better. So you see, it was only one parameter which has been adjusted here, and such a much nicer blot has been produced. And here comes some advice for beginners. First, focus on recording correct band before you multiply variations of conditions. I have seen it over the years that people struggle and change membrane, change buffer, do all different things without some kind of helicopter view on all this. Because if you consider all these parameters at first and plan an experiment, and the target goal first is to see a band of correct molecular weight. And if there is a lot of background around it, it doesn't matter because you can optimize it later. So for example, I would do like this, that I would only use wild type and choose the mutant. I would load quite a lot of protein per well. I would fractionate if necessary. And I would incubate primary antibody overnight. Yes, the dilution of primary antibody is 1 to 500 means that primary antibody is quite concentrated. But it means that I'm overloading. I just want to see if there is a proper target band. And later on, I can work with the protocol and refine it. And here comes an advice for experienced users. Look over the protocol which you use. One and the same protocol does not fit all antibodies. This is the most common mistake, that we ignore molecular weight of a protein or its cellular localization, and we generate a lot of work for ourselves, which would not happen if one would look through and consider what is my target protein, where is it expressed? Is the protocol which we have in the lab suitable? So before optimization, it can look terrible. After optimization, it may look really nice. If you have such blots, I suggest that you don't panic. I am always eager to help and answer questions. Even if you use an antibody which is not from Agrisera, but which you received from a colleague, for example, based on my experience, I can try to help you. For those who are shy and don't want to contact us, we have technical blog where we are posting about different topics with pictures. For those who would like to be updated on technical tips, videos, we have our newsletter, which is sent once a month only, and you can sign off at any time. We also have a Western blot resource, which contains a little bit of short videos, and you can walk through it anytime you want. You have received our Western blot guide as PDF and also a printed copy, which you could have in a lab if you find it useful. And also a reminder that we have educational resource here with images extracted from the posters which you have received, which you can use in your presentations or publications. And poster 6, you have received 5 posters, and there is a poster 6 coming, and I guess you are now understanding about which topic it's going to be, because it's going to be about small RNAs. And for those who are interested, you can sign up on our website to receive the hard copy when it's available during the summer. I would like to wish you good luck with your blots. As you see here, it is possible for high abundancy proteins like PSBA, photosynthetic protein. We have Argonaut here, and we have a catalase, which that blot shows actually these extracts are from all different plants, so-called garden blot. You will have a second chance to do the quiz, which is different. It's different type of questions, and for those who are interested, you can do it after that workshop. As I said, there will be few winners, not only one. And I would like to thank you for your attention with this view of from our beautiful surroundings during the Swedish summer. And if you have any additional questions, they are most welcome. I will leave the slide with the quiz. I can also send a link to the quiz in the chat.
Thank you, Joanna. It was great learning experience, and if you have any question, please feel free to post it in the chat. Otherwise, please join me in thanking Joanna one more time. And, yeah, go ahead.
Yeah, I would just paste the link to the quiz. I maybe stop sharing too fast.
Yeah,
so
that's fine, and I can also just circulate the quiz, email them to the entire participant list, so that should be fine too.
Yes, absolutely, and I hope that you enjoy the quiz and that you have got to know a little bit more about the Western blot techniques, maybe things you didn't know about, and you are always welcome to contact me if you have questions.
I think we have one question here. It's from Trishta Lee. She's asking, like, maybe also somehow, oh, okay. She's asking to give out Joanna's contact info. Is it okay, Joanna, if I give your email so that they can contact you as per requirement? Okay.
Absolutely. And also I will send you PDF of the presentation, which you can share with everybody.
Yes, yes. And thank you. I know it's pretty late in Sweden right now, but thank you for your time. It's truly appreciated.
Oh, thank you. You're all most welcome. It's only 6:45 and we have no nights. So
okay.
It's no problem at all. And so if you have questions afterwards, you're most welcome. And thank you for your attention.
Thank you. Thank you. Thank you.